talk will be focused not on diabetes itself, but on one of the major complications of diabetes, which is the diabetic foot ulcers, due to the fact that in Cuba, since we have a high standard of education and health, so we are facing the same problems of diseases related with aging. So we have cardiovascular diseases, we have diabetes, we have cancer. So we are working very hard in trying to fight, and to solve these problems. But before we get into more details, I would like you to know a little bit about our main biotech institutions in Cuba. The West Havana Scientific Port, which is a biocluster devoted to life science and biotechnology in Cuba, is formed by 53 institutions at the present. All of them engage in different particular missions, development of vaccine, diagnostic means, devices, and so on, but integrated all together to develop biotech in Cuba. Some of the most important ones are shown. For instance, here you can see the Center for Genetic Engineering and Biotechnology, where I belong to, the Center for National Research, which is the pioneer center of research in Cuba, the Fila Institute, which has to do with research, vaccine research and development and production, is well recognized worldwide. You are going to see and to hear a little bit more this afternoon. The Center for Neuroscience, they do technologies and devices and develop technology and devices for the brain function study. The Center for Molecular Immunology we have to do or is devoted to develop monoclonal antibodies and vaccines against cancer. For instance, other institution, the BioCen, which is a big production facility in Cuba where we finish all our raw material there. And there is 2 other centers which are engaged to develop biotech in Cuba. Among these institutions is the Center for Genetic Engineering and Biotechnology, inaugurated in July 1986, which is one of the most important and biggest research production facilities at the West Havana Scientific Port, endowed with the state-of-the-art equipment and staff to close the cycle from biological research, which means from biotech product and process development to production, registration, and commercialization. The CIGB and its commercial branch, Heber Biotech, already produce a positive impact in Cuban society and a positive cash flow from sales to more than 40 countries at the moment. Here we show how the commercial activity has grown up at the CIGB. Until the year 2000, we had just a few products, as you can see, mainly the hepatitis B vaccine, some interferons, and some diagnostic means. Today, we can show near 20 products and technology in the portfolio, most of them innovative products with a huge registered and commercial activity in different territories. One of these innovative products is Heber Product P, a unique Cuban biotech product for the treatment of diabetic foot ulcer to reduce the risk of limb amputation, particularly for end stage of the diabetic foot ulcer. This product has been 100% developed by the CIGB in collaboration with the National Institute of Angiology and Vascular Surgery in Cuba. As you can see here, that this is the diabetic outlook in the world. So you can see a high prevalence rate by countries and also regions, and there is an estimate at the moment of around 200 million diabetics worldwide, and it's expected to increase by 2025 to more than 300 million, mainly due to sedentary lifestyle, increase of life expectancy, diet, and so on. So it is also stated that from the total population of diabetics, between 10 to 15% develop a diabetic foot ulcers, at least one in their life. And from that number, 10 to 15 also end with an amputation. So the message is clear. The more diabetes we have, the more diabetic foot ulcers and amputations develops. Experimental evidence at the CIGB in the 90s in the rat model of total allotomy of the sciatic nerve demonstrated that the recombinant EGF infiltration not only contributed to the morphological and functional restoration of the injured nerve, but also delayed and attenuated significantly the appearance of those trophic changes, like ulcers and necrosis of the fingers. The effect of recombinant EGF on the restoration of peripheral nerve subjected to severe traumas offer us the theoretical basis for a possible treatment of the chronic ulcerative injuries of the skin. Then, at the CIGB, we developed a product, the HebeProp-P product, which is a supported product by a patent application of use of a pharmaceutical composition and a unique method of treatment, containing EGF recombinant 1 in an injectable formulation which is administered by infiltration for the treatment of diabetic foot ulcers to accelerate the healing of deep and complex ulcers. This preclinical evidence showing the efficacy of EGF in the neurogenic ischemia and protection of peripheral tissues have been published. On the rationale that EGF could enhance healing of chronic wounds following repeated local infiltration, we postulated the hypothesis that intralesional infiltration of HebeProp-P can enhance local cell survival and stimulate the healing process of diabetic foot ulcers, but stimulated the cell that participates in the mechanism of ulcers, like keratinocytes, fibroblasts, and endothelial cells as well. Therefore, in 2001-2, a phase 1 clinical trial in 29 type 2 diabetic patients to whom the decision to amputate had already been taken was carried out at the National Institute of Angiology and Vascular Surgery. Patients were classified as ischemic infections or neuropathic fit with ulcers bigger than 20 square centimeters, having chronic diabetic wounds recalcitrant to heal with scar or no granulation tissue at all. HebeProp-P therapy succeeded in healing and preserving the fit in 17 out of 29 patients. So the most remarkable results in terms of efficacy here for a therapeutic niche like this, where there is no other choice but amputations. And the other important thing here to highlight is that the level of recurrence was really low in this study. These results gave us the possibility to move on a phase 2 clinical trial in 41 patients between 2003 and 2005, a controlled double-blind, randomized, and multi-center phase 1-2 clinical trial conducted in Cuba also for the safety and efficacy assessment of the HebeProp-P. Also in patients with Warner grade 3 and 4. The trial was performed to test the EGF in dose of 75 or 25 micrograms of EGF through intralesional injection 3 times per week between 5 to 8 weeks, along with the standard wound care. This is the design of the study, also the inclusion and exclusion criteria followed during this study. The hypothesis was confirmed in this case. In the follow-up stage, as you can see in the picture, upon 1 year of follow-up, wound healing measured by the complete closure of the wound was observed or achieved in 53% overall among both groups of doses, and we were able to rescue from amputation 66% of patients that were diagnosed in most of 90% to be amputated.